Learning Outcomes, Subtopic 5.1.2a. 5.1.2a Illustrate to explain Krebs cycle. Krebs cycle is discovered by Hans Krebs in 1930s. Krebs cycle is also known as citric acid cycle or tricarboxylic acid (TCA) cycle. In this cycle, acetyl-CoA is oxidized in a series of reactions occurs in the matrix of mitochondria. Acetyl-CoA combines with a 4-carbon molecule, which is oxaloacetate or OAA, to form a 6-carbon molecule. The 6-carbon molecule is then oxidized and 2-carbon dioxide molecule is removed to restore OAA. OAA is then recycled and bind to another acetyl-CoA. Steps in Krebs cycle. The first step, acetic group of acetyl-CoA joins with oxaloacetate to form a 6-carbon molecule citrate. This reaction is irreversible. In the second step, citrate undergo isomerization to form isocitrate. A water molecule is removed from one carbon and added to a different carbon. In the third step, isocitrate undergoes oxidative decarboxylation. It removes one carbon dioxide molecule to form alpha ketoglutarate and removes two hydrogen atoms to NAD+ to form NADH plus H plus. In the fourth stage, alpha ketoglutarate undergoes oxidative decarboxylation. It removes one carbon dioxide molecule which converts alpha ketoglutarate into succinyl group, and it removes two hydrogen atoms which reduce NAD+ to NADH plus H plus. One coenzyme A joins with succinyl group to form succinyl-CoA, which is a high-energy molecule which have unstable bond. The fifth step, when the high-energy bond between CoA and succinyl is cleaved, energy is released. It is used to bind phosphate organic in the matrix to guanosine diphosphate to form GTP or guanosine triphosphate. Then GTP transfers its phosphate group to ADP to form ATP. Substrate level phosphorylation occurs. Succinate group is converted into succinate. The sixth step, succinate undergoes oxidation. It removes two hydrogen atoms which reduce FADA to FADH2. Succinate is converted into fumarate. The seventh step, a water molecule is used to convert fumarate into malate. The eighth step, malate is oxidized to oxaloacetate. Two hydrogen atom is removed to reduce NAD+ to NADH plus H plus. Oxaloacetate is regenerated and bind with other acetyl-CoA molecule. The cycle continues. Summary of Krebs cycle. This is the Importance reaction in Krebs cycle. 2-isocitrate is synthesized into 2-alpha ketoglycerate. 2-alpha ketoglycerate is synthesized into 2-succinyl-CoA. 2-succinyl-CoA is synthesized into 2-succinate. 2-succinate is synthesized into fumarate. And the last one, 2-malate is synthesized into 2-oxaloacetate. The first reaction will produce 2-NADH. The second reaction will produce 2-NADH. The third reaction will produce 2-GTP, which will then proceed to 2-ATP in substrate level phosphorylation. The fourth reaction will produce 2-FADH2, and the last reaction will produce 2-NADH. In summary, substrate level phosphorylation occurs and produces 2-ATP, while 6 NADH molecule and 2 FADH2 molecule will go to ETC. Please refer to your tutorial notes. In Krebs cycle, there are no ATP investment, but oxidation occurs at step 3, 4, 6, and 8. Decarboxylation occurs at step 3 and 4, and substrate level phosphorylation occurs at step 5.